to read this review paper for better understanding of the multi-omics data. And then there are some advanced or most relevant part in big data analysis, which most of the people prefer to go for analysis at this level, that is called functional analysis. And the basic thing in functional analysis is gene ontology analysis, where we go for molecular function, biological process, cellular component analysis. So this gene ontology gives 3 GEO terms, that is called molecular function, biological processes, and cellular components. The next part is pathway analysis, and also it also includes like differential expression analysis, which is the first thing after gene analysis. And then we go for functional analysis for the differentially expressed genes. And the tools mostly used are or accessed are David, GSEA, GSEA, CAG database, GEO databases, Panthers, and more, which we can summarize in the next part. Then it is another level of analysis that is called network analysis. Here we study the protein-protein interaction network, gene regulatory networks, co-expression networks, and more things. mRNA networks are also these days studied most frequently. The tools, common tools are Cytoscape, String, WGCNA, and there are some more databases, for example, FunCup. Then it is like other protein network databases. Then here we are again going to summarize some of the softwares like genomics study, mostly GetK, PWA, STAR, and more softwares are used. For epigenomics, bisulfite sequencing, chipseq sequencing analysis is carried out. For proteomics, MaxQuant, Proteome Discover are used. NCBI Ensemble and UniProt are the common databases. Then if you want to go for analysis or implementation, you can go for implementation in cancer multiomics profiling, diabetes multiomics profiling, or any kind of infectious disease multiomics profiling. There are also applications in plant sciences. So some kind of metagenomics analysis, and then metatranscriptome analysis. Some things are like, some more things like microorganisms could also be studied. And future directions like advances in single-cell sequencing, real-time data analysis, ethical considerations, and data privacy. In conclusion, we can summarize the key points, which are like to learn the basics, what we include in multiomics data and the importances of these data, and how to analyze these particular multiomics data for a specific type of goals or diseases. And most common softwares for such multi-omics tools, of course, the listed softwares are not the only softwares, but these are some of the helpful softwares like ShinyGo, Panther Database, David, GENZ, Enrichment Analysis, GSEA, EnrichNet for the GET pathway, and Gene Ontology Terms Analysis, GEO, like Gene Ontology Database, KM Plotter for survival analysis, NCBI, and more. Finally, in the next part, we will go for some of the detailed implementation and